So welcome to part 3 of the annotation worksheet solutions, and this time we are working with the solutions to the Apollo one, which is a little bit more hands-on, and throughout this I will be pulling up Apollo in certain instances and showing how to do certain things if you need a refresher. But just to get started on the first question with something a little bit more conceptual, just to understand what exactly Apollo is. So Apollo is a genome browser and editor. Genome browser, from the first worksheet, we know the definition of a genome is seeing the full genetic complement of a particular organism. So in our case, D. citri. By using Apollo, we are able to go in and view the entire sequenced genome. So here we have a gene model of this here unknown protein. It's just a random one I pulled up. But we can actually zoom in to the base pair level. So we're able to see the nucleotide specifically within this organism, and by further extension, the different possible proteins that are being made. So being able to view this entire genome, we're able to zoom in on the very specific nucleotide level, but we can also zoom out and this is only one scaffold, but we can see all of these different gene models. Each of these is a different mRNA transcript that encodes for a particular protein. And this is only on scaffold 1. We have all the different scaffolds that we can look at. So there's lots of different genetic information in D. citri. So beyond just being able to view the genome, we can actually make specific edits if we wanted to. So I don't think I've showed you this, but let's say that my evidence that I'm consulting, maybe it's sequences from other orthologs if it's a very highly conserved gene, or maybe some splice site is occurring that by changing a particular nucleotide in the code that you think was sequenced incorrectly, you can actually make a substitution. So I could change this A to a C or something of that sort, or I could insert perhaps a missing nucleotide if it's causing some sort of big error. So beyond just viewing the genome, we can actually make edits to the individual code. So that's what it means to be a genome editor. Yeah, so we can modify these precise locations in the genome, and also more commonly is we'll be making changes to the mRNA transcript of these gene models, consulting RNA-seq or evidence from other orthologs to come up with the best version of the RNA transcript. Something that's unique about Web Apollo is that it was the first web-based genome annotator that is instantaneously updated and collaborative. And by that I mean, we can have the 13 annotators that work at IRSC all go into Apollo at the same time and make changes, and it gets updated. Web Apollo was the first sort of program to do this in this particular way back in, I believe 2013 is when it first got released. So moving on to the second question. What is a gene model? So the gene model specifically is kind of referring to this mRNA transcript and its visual depiction in the genome annotator. So here for the screenshot, this is all one gene, this particular cytochrome P45. But for this particular cytochrome gene, we have all these different versions of the gene model. And the gene model is this depiction of the mRNA transcript that includes information about the splice sites, the exons, the introns, with your introns being the black lines and the exons being your colored boxes, and the UTRs, which are your white boxes. So this sort of depiction of the mRNA transcript from the DNA, which we can then, click on and get information about the protein. So that's what we do when we get sequence and then get the peptide sequence and collect that. Fundamentally, we're working at an RNA level in the annotator box and looking at these gene models. All right, next question, number 3. What are reads, contigs, and scaffolds when talking about genome assembly? So this will give you a little insight into how the genome was sequenced and inputted into Apollo. which I think is pretty fascinating. This is what Dr. Surya Saha works on. He works on sequencing and this sort of pipeline type stuff. But whenever the DNA is sequenced, what happens is we have an individual chromosome, but it's not sequenced all at once. It's really long, and it's difficult to sequence something that's potentially hundreds and hundreds of thousands of base pairs long. So what they do is they sequence it little by little, and then they take the little pieces, and that's what we call a read. So there's maybe a particular fragment of the DNA. So let's say this is like 150 base pairs long or something. And what they do is they make a bunch of different overlapping copies of sequenced information here. So these 4 reads are coding slightly different. This has more of the beginning part, here's more of the middle, here's more middle, and this is sort of the end. So these reads overlap in places. So where they're overlapping, the code is the same because they're coding the same part of the chromosome. So this is unique code for this first bit, and then here in this area, these 3 reads are overlapping and it's the same code. And then here, these 3 are overlapping, and then here's some unique code at the end. So to kind of show you on a smaller scale, let's say that we're coding a 6 base pair long segment of DNA. So each read is 4 nucleotides long. So in the first read, we get A, T, C, G, and then we have a second read that codes T, C, G, A, and then a third that is C, G, A, C. So then once we have these different reads, the computer program is able to recognize the overlapping parts. So it's able to see that the TCG is overlapping with the TCG of the next read. By having a bunch of these different reads, we are then able to line them all up and then get the fuller, lengthier sequence. So we're able to get the full ATCGAC. Reads are a lot longer than this usually. This was just a kind of small scale example, but it kind of shows you how the reads form and overlap. So what happens is each of these would be considered a read, and then this would be considered a contig. So a contig is sort of the smaller portion of the chromosome that is assembled based on reads. But the contigs are only still fragments. They're sort of assembled fragments based on the reads, but there's still these gaps between the different contigs. So then they try and close these gaps of the contigs, and when they do that, we're able to get a scaffold, which is sort of this more complete version of the chromosome. And then all of the scaffolds together create the chromosome. So it's sort of this very fractured and multi-stepped process that we go from the smallest and we try and put them and piece them together like a puzzle until we get sort of a, as good as we can, depiction of what the chromosome actually is. So in Apollo, it's not particularly that each scaffold is its own chromosome, maybe, but it could be that 3 scaffolds all give the complete picture of a particular chromosome, but they're just trying their best to sort of give representation of the genome, and maybe it's not perfectly put into, here's chromosome 1, here's chromosome 2, but there's still chunks of the genome. So each scaffold is like a very large chunk of the genome. All right, moving on to the next question. Number 4, what is the difference between 2.0 and 3.0? 2.0 is just the older version of the genome. So when we're talking about annotation and validation in the next question, which I'll get to, in 2.0, we had an older version of the genome based on the sequencing data that was available at that time. And when the sequencing was done computationally, we had annotators that have to go in and ensure that the gene models that are being predicted are actually true. So we make manual annotations, and those older annotations are done in the 2.0 version of the genome. When the annotations are in the annotator box, they're not yet officially incorporated into the canon quote-unquote genome. Like there's the sort of official, they call it OGS, the official gene set, like the canon version. And that's why we can go in and make these manual adjustments, drag things into the annotator box. It's not set in stone, right? Like you can put something into the annotator box and you can delete it, and it doesn't immediately have an impact on what is in the official gene set at the moment, right? But whenever the genome updates officially and goes from 2.0 to 3.0, all of the stuff that was put into the annotator box gets incorporated officially into the next gene set. Again, it's incorporated along with any new sequencing data that's also incorporated, so it may not look exactly like what you annotated, but the idea is that your changes that you made get incorporated into the new genome. So that's what happens between genome version updates, to put it simply. Next question, number 5, the differences between annotation and validation. Annotation is just the name for the process of using the genome editor to view these computer predicted gene models and make changes to them based on its available evidence. So whether that be RNA-seq or using orthologs from Blat, things like that, we try and create the best version of the gene. That's just the name for this whole process that we're doing. Validation is considered a subtype of annotation work, specifically while you're looking at older annotations. This is what we're doing when we're looking at the 2.0 and comparing it to the 3.0. That's what validation is. So really, annotation is just what we're doing in Apollo to improve the gene models. And validation is that annotation, but specifically trying to reconcile it with the previous work that was done in the old version of the genome. And the reason that we have to do this is because whenever new sequencing data becomes available, we sort of have to look at the work that was done in the previous version of the genome, look at how it's changed in the new official gene set, and make sure that what's present in the current version is in its best form. So question number 6, what is Blat? How is it different from BLAST? They're both very similar. In the last worksheet, we went over what BLAST was, so I won't go over it too much, but it stands for the Basic Local Alignment Search Tool. But Blat, it actually stands for BLAST-like alignment tool. While BLAST takes your query and then it compares with other full sequences in the NCBI database, for example, and there's other databases and other things that BLAST aside from just NCBI. But when you're in Apollo and you're blatting, it is similar to BLAST in that it's comparing similar sequences, but where it differs is that it's looking at much smaller segments. So if you input, let's say, a peptide sequence that's 100 residues long, the Blat will find sequences of length that are maybe around 40 bases long. And what this does is it kind of scans the whole genome, or perhaps the whole scaffold if you didn't select all genomic sequences. And it's just finding little segments here and there that match your query. In short, though, Blat is what we do when we hit this search bar here. When we're inputting things here, we're looking at Apollo, we're looking particularly in the dCitry genome in Apollo, versus BLAST, which is looking at a ton of different sequences from a ton of different organisms in a larger database. So to examine this Blat, what is ID, score, significance, and identity, and why are each important to consider? So just to go from left to right, ID, this is the ID for the scaffold. So we see DC3, which is dCitry 3.0, DC 3.0, dCitry 3.0, that's the 3.0 version of the genome, and then SC, which is scaffold, and then it tells you what scaffold it's on. So SC6, if we went into Apollo, if we wanted to go to that scaffold, we would go to DC or SC06, which is the scaffold 6. So the start and end. So this tells you the base pair location within a particular scaffold that your hit is in. So here in Apollo, if we zoom out as far as we can in the scaffold, we see the sort of full range of where the scaffold is. So it starts at 0 and it's going over to about 40 million here. Yeah, so here we're at the very end of the scaffold, and here at the top of the bar, you can see where your relative location in the scaffold is. So we can see we're all the way here at the end. So since this is scaffold 6 in Apollo that we're on right now, and this here is talking about scaffold 6, this is saying this is towards the end of the scaffold. So this kind of just lets you know positionally where in the particular scaffold you're looking at it occurs. And this is really useful to tell if your hit is unique. Just by glancing at this, I'm thinking that all of these 5 hits exist in the same gene model, just by looking at the start and stop end locations. So perhaps this is a unique gene model. This is unique because it's at 42. These two here, they're separated by 100,000. That's actually quite a lot. So these might be different, but they could be the same also. This one, that's 3521, and this one is also 3521. So these two are probably the same gene model, but this one here might be unique. But you'd have to go in and double check. It's always good to double check. And then again, this 38 million on scaffold 1 very well might correspond to these up here. So just at a glance, I'm seeing 1, 2, 3 different gene models possibly here, or more, but at least 3. But again, you want to double check. All right. So to talk about the differences between score, significance, and identity, first let me just talk about significance. This is pretty similar to E-value in BLAST, but it's just talking about how statistically significant your search result is. The closer to 0 you are, the more it matters. So all of these results here at the top that have a score of 0 for significance, that means they're worth paying attention to. This last one here that has a significance of 36,000, don't pay attention to it. It's not worth considering. Typically, I just look at everything that has a significance of 0 because those are results worth considering. So to talk about score versus identity, identity is a particular rating out of 100 percentage-wise of how similar the query to the hit is. So since Blats are usually doing small sequences of around 40 bases, let's say, for example, this first one is 40 BP long. It gets a score of 180, and it's 100% identical. Down here, though, it's 100% identity, but the significance is much worse. It's not closer to 0. So it's saying it's not as statistically significant, and the score is much lower as a result. But score is kind of just this weighted grade of the similarity. All right, next question. So if when doing a Blat, I don't select all genomic sequences, what happens? And what do I do if I try blatting a nucleotide if I've input a protein? So going back here, this blue check mark here for all genomic sequences. As you can see here, we have a bunch of different scaffolds that pop up in the hit results. If I didn't have this check mark checked, it would only scan the current scaffold that I'm on. So here I don't have it checked, and I'm in scaffold 6, so whenever I put in a sequence and then I search, it is only going to scan along this one scaffold. So if you're meaning to search for the entire genome, then it's really important that you have that checkmark checked. And then here I've inputted a protein, you can see, because of all these different letters as opposed to just the 4 letters that you would find in a nucleotide sequence. But if you inputted a nucleotide sequence and tried to blat protein, or vice versa, it just wouldn't work. It would probably just sit there loading for a long time, and then after wasting your time for 30 seconds or so, it would give you an error. So make sure that you're using protein with protein and nucleotide with nucleotide. Question number 9. What is more specific, a peptide blat or a cDNA blat? This should be a pretty simple question if you sort of understood the concepts of the first worksheet when I was talking about the wobble position and cDNA specificity. So if you're a little shaky on that, go back to that video and watch that particular question where I go over it. But essentially, when looking at a codon usage chart, you can see that multiple different DNA or RNA sequences, in this case RNA for this particular usage chart, will encode for the same amino acid. So these 4 different sequences of RNA all make for serine. Back in that first worksheet, I made you guys give 3 different mRNA sequences that encoded for the same protein sequence. So in that sense, cDNA is a lot more specific because you can have the exact same identical peptide sequence, but the cDNA can be different. So the cDNA is going to be 3 times longer, if not more, than your peptide sequence. To look in Apollo here, looking at this little gene model here. The peptide sequence is 121 versus the cDNA sequence is 1677. So it's the same region. It's the same length of the genome that it's being examined. Like, both of those are just talking about this one particular region here, right? But the cDNA is sort of this more specific and longer-winded version. So an example of when you would use a cDNA blat over a peptide blat is particularly if you're trying to find if there's like an artifact duplication or something. But artifact duplications, in short, are sort of errors in the sequencing where a region of the genome got copied. So there's 2 gene models present that are identical where there should only be 1. So you can only verify that they're copies by looking at cDNA similarity. So question number 10, explain the location tag, why it's useful. Here in Apollo, circle the location box. It's important because when you're looking at a particular gene model, you're at a particular magnification view that you're looking at something very specific, the location tag will take into account the same magnification level. So say I'm looking at this one particular exon and I'm saying, oh, maybe these should connect, maybe they're part of the same protein. What I can do is zoom in specifically to this location and say, oh, this is where I'm talking about. So then I can take the location tag and then put into my notes, at this location, I think that perhaps these exons should be merged together into the same gene model. And you can put that there. That way, if you're working collaboratively with someone else and you don't have time to work on it, but you can delegate it to someone else, they can go in later. And if you took good notes, they should be able to find what you're talking about. Magnification is really nice in that regard, that it helps if other people are working with you, but it also helps you easily get back to something that you're working on. So it's very useful in that regard. Question number 11, what is an AHARD predicted name? How reliable is it? I included a link to a publication that goes into a little bit more depth if you're feeling curious as to what and as to how this is made. But it stands for this automated assignment of human readable descriptions. So essentially you'll have this peptide sequence or something like that, and the AHARD is sort of this program that is trying its best to mimic the sort of decision-making process of a curator or annotator. So what actually happens is it does all these blasts. It scans through like a blast table. It finds all of the sort of common names for a particular gene. It's sort of like what we did in Actin, where I had you look at what the different names were coming up as. So some names aren't useful, like it'll say like hypothetical protein or unknown protein or putative protein. So it sort of has in its code that, oh, those aren't useful names, so I'll get rid of those. And then it'll say, okay, which of these is reliable evidence? How common is it popping up? And then based on these sort of blast results, it will then computer predictedly name the protein in Apollo. So that's what happened when we're looking at this protein pangolin. A computer program actually went through blasts and made its best guess on what this protein was named. And a computer can only do so much, right? Like, they're not perfect. That's why sometimes you'll make changes to the name, or you'll make the name more specific. I've worked on a gene model called protein kinase C where it just said protein kinase C. But upon doing further research, I found that it was protein kinase C I isozyme, or something more specific. So then I can go in and then rename it. The computer program wasn't smart enough, I guess, to sort of see that nuance, but as a curator, you can go in and fix that. Number 12, next question. Which ORF is being used in this exon? What is the directionality of the gene model? And I asked you to circle the start or stop codon. This is kind of small and very subtle, but here in the corner, you can see that there is a little arrow here pointing in the leftmost direction. So what this is saying is that the gene model is reading right to left. So it's reading backwards in this direction. So which ORF is being used? Whenever something is reading right to left, that means it's using the bottom three of these reading frames. And then the color is corresponding to which particular one of the three. So since it's moving in this direction, which is what the arrow says, we know that we can look at the bottom three, the bottom half. And then since it's this color, we know that specifically out of these 6, it's this blue bottommost open reading frame that is being considered. Then I asked you to circle the start or stop codon. I included this as a nice example because there was both a start and stop codon. So if you didn't know which direction the gene model was reading, if it started on this end, you could be forgiven for thinking, oh, this is the start codon, it starts this, but hey, there's also a stop codon. So since it's reading in this direction, if you zoomed out here, you would actually see that the gene model was ending here. And this is in fact the stop codon for this particular gene model. I thought this was a pretty fun question. Hopefully you got it. Next question, number 13. In this fragment of the gene model, label the UTR exons and introns present. So this should be very simple, especially if you saw the solutions video for the first worksheet where I talk about that. But the exons are the colored boxes. I have them labeled here as exons. This here is the UTR, and it's reading 5' to 3'. So we know this is the 3' UTR since it is on the end here. Actually, oh yeah, this is 10170 2.0. Yeah, these are the same. Haha. So zooming out, this is actually here. So you can see that since this was the stop codon, this was the end of the gene model, just like I said. So it's reading 5' to 3', right to left, using this bottom ORF. But yeah, so the gene model ends here. That means that you know where the arrow is pointing at the end. That is the 3' UTR. And if we saw the rest of the model over here, you would see the 5' UTR. The introns is what is spliced out during mRNA splicing. And those are the black lines. That's the introns. Simple enough question. Now number 14. You don't have to be super concerned with all of the depth of Illumina sequencing, but I just kind of want you to have an idea of how it works. So here's a YouTube link to a quick explanation of how Illumina sequencing works. It's only a 4 to 5 minute video. It's pretty simple. The first 3 minutes talks about the setup for it, and I sort of outlined the steps here for the setup. Essentially, the setup phase, which is comprised of these steps, and the first 3 minutes of this video goes over, is getting it ready for sequencing. But what's more interesting is this sequencing by synthesis, which I'll show here a little bit. You start off with sort of this primer added on to this adapter at the end. And the way that sequencing happens is they take these nucleotides, and each nucleotide is fluorescently labeled. So we have thymine with blue, cytosine with red, guanine with yellow, and adenine with green. So they're sort of all here in solution. So they'll be subjected to the strand that you're trying to sequence, and they won't bind unless it's complementary. So C will come in, the G will come in, the A will come in. None of those bind, but the T comes in because it is complementary to the adenine. So then once it binds, since it has this fluorescence tag, it's going to be shot with a laser. And this laser is going to be excited and fluoresce a particular color based on the color of the thymine. So all thymines are going to have this blue fluorescence. So based on when it's shot with a laser, it fluoresces a certain color. Then based on the different color that's excited, you know what the sequence is. So this is how they go through and sequence this length of genomic information, is by literally adding on nucleotide at a time and then exciting it with a laser, and the color is giving you the sequence. So that's Illumina. It's a bit of a detailed process, but what's important is understanding that all of this is being done little fragment by fragment, right? When I was talking before in the previous question about reads and contigs, so these reads, maybe this one read, like this whole entire flow cell process where a bunch of stuff is sort of sequenced in unison, this is all done for like one read, right? So all these reads are being sequenced at this one location, and this process has to happen for every single read that's going on. Each of these is sequenced in this particular way, and all of that information is added up to get, our scaffolds in the end, which is kind of interesting. But again, it's just sort of reiterate on this part that this is being done in fragments, and because of this, gaps form. Perhaps you just didn't get proper coverage, and maybe there's a gap between a contig, or the scaffolds don't fit quite together right. Like there could be anything where just like gaps occur in the Illumina sequencing. So it's just good to be aware of that, and we can actually see that happen in Apollo. So this next question, number 15, what happened here? This is a gap. This is a gap in the Illumina sequencing. I think each of these individual boxes is a read, and so all these reads together would be a contig. So we're looking at one contig here, and then another contig, and these contigs just didn't, there wasn't any genetic information lining them up together. So that's why we have this gap thing. So this is a gap in the Illumina sequencing, probably due to the contigs not being connected. And what happens is this is just having some missing data in the genome. So looking here at the context of this gene model, this gap is kind of occurring in an intronic region. And in the first worksheet, we kind of talked about how introns are more important than most people give them credit for. They have important sequences that help with splicing close to the exons, and they have the branch point A. Like there's a lot of stuff going on in introns that help the gene get expressed and create the correct protein. So it's important that we understand them. So having this gap here, it's just important to note. And it may sort of explain why you may get an error when you're trying to annotate something. Like if you blast your gene model and there are exons missing, like maybe say when you look at orthologs, there are exons present here. But when we look at our gene model in Apollo, it's just intron. So if that occurred, that could be explained by this gap in the sequencing, and you would have to account for that in your notes. Maybe there's nothing you can do about it, because at the end of the day, if the sequencing data just isn't there, there's not much you can do to fight it, but you can at least make an observation about it and include that in your notes, which is part of just being thorough with your work. So compare and contrast duplication and artifact duplication. Touched on this a little bit earlier, but this is actually a document that Yasmin gave to me that is based on a conversation that she had with Chrissy, where she was kind of explaining it. You can pause and sort of read through. This is just between their conversation. But essentially, an artifact duplication, if you're in Apollo and you see this one gene here and you blat it and you find a completely identical copy somewhere else in the genome. You compare the cDNA data, you pairwise blast them, and the cDNA is 100% identical. They're in completely different locations. You can be pretty safe to assume that that's what's called an artifact duplication. And that's when the sequencing data, the assembly of the different contigs kind of got messed up. Maybe accidentally the gene model was copied and put somewhere else. So what happens is, if you find some sort of artifact duplication like this and you're pretty certain, you have to list it for deletion. So in the annotation master document, there is this link for listing artifact duplications, which I will pull up here. Yeah, at the bottom of the document, at the bottom tab, there's a thing that says to remove OGS 3.0, and you go to this your worksheet. You just scroll to the bottom, and you can see here's an artifact duplication that I listed earlier, where this is the one I want to delete. I have my name, and for the reason of removal, I say this is an artifact duplication, and this was a case where it was a chimeric duplication, where two gene models were merged together. So I confirmed that each individual gene model was just an artifact duplication. So I put all those down, and these will eventually get listed for deletion later. So that's what you do if an artifact duplicate is present. You can find the link to this document on the resource master document. And what is the difference between that and a regular duplication? So a duplication refers to a naturally occurring duplication in the genome. So let's say that in the organism in nature, a region of their genome got copied and pasted somewhere else in the genome. And this tends to happen. If you've ever heard of something called pseudogenes, this is actually pretty common, where a functioning gene will exist. The functioning gene, something will happen, it'll get duplicated accidentally, probably during some error in replication during mitosis or meiosis. And having this extra copy of the gene is nice because the working copy is able to function normally, but then the duplication is free to accumulate mutations and change, and perhaps they can gain new functions. If you're interested in pseudogenes, I'd highly recommend looking up. It hasn't really been a topic that is super important to annotation, so I won't bother going into it, but it is fascinating in my opinion. But yeah, so duplications are like copies of genes that are actually supposed to be there versus artifact duplications, which are computer errors that need to be taken out. Moving on to question number 18. So how would you switch the direction of a gene in the annotator box? So here, for example, I want to switch the direction of this, so you just highlight the whole gene and click move to opposite strand. So now it moves it to the opposite strand, and usually this will create a bunch of splice side errors. But occasionally you might want to do something like this, and you can always change it back by doing the same thing. How would you split the following two exons? Here, so just this random gene model to use an example. What you do is you select one exon, you hold down the shift button, you select another exon, you right click, and then you split. And this splits the gene into two different models. Explain how do you merge the following two exons. So you click one, hold down shift, click another one, and merge. Is it possible to merge these two exons? If not, what steps would you need to take? So note that these are in opposite directions. This one's reading left to right, and this one's reading right to left. In order to do this, here I'll show you, I would split these, then I would select one of them, and move to opposite strand. So now these are facing two different directions. If I went in and tried to merge them, it doesn't give me the option. In order to merge gene models that are facing opposite directions, you need to highlight the whole thing and move to opposite strand. And then you're able to merge them back together. All right, number 22. What is RNA-seq? What is read depth? So RNA-seq, to keep it as simple as possible, is this way to quantify gene expression by looking at how much RNA is generated from a particular gene. By this I mean you have a gene made of DNA, transcription occurs, and mRNA is made. Some genes are expressed more than others. This means that a gene will make more copies of RNA than other genes. Not all genes are expressed at the same rate. Some genes you need to be constantly expressed. Examples of genes like this are maybe genes that encode histone proteins. You always need histone proteins. Or you need ribosome protein. You always need ribosomes. So genes that encode these are much more active. And you can actually see how active a gene is in being expressed by the amount of RNA that is transcribed from it. So what RNA-seq does is it takes the RNA, turns it into cDNA by reverse transcriptase, and this I talked a bit about in the first worksheet if you want to refresh on that. And then this cDNA can be analyzed and quantified. So in Apollo, pulling up the XY plot, this gauge here gives you sort of an idea of how many RNA reads there are. So you can actually see at this particular location in the gene, and it has a scale here out of 80, that gives you a relative amount of expression. So similar to how Illumina works, it doesn't just sequence the RNA all in one go. The cDNA is encoded in fragments and then overlapped. So this creates reads, just like we talked about in Illumina with these reads of DNA, but instead the same thing happens with RNA. So same with Illumina, the read depth means the more reads that are overlapping. So here we have a read depth of 3. There are these 3 reads that form this one contig. But if you had hundreds of reads, that means that you're more confident in this contig, the sequence, this compiled sequence. The more read depth you have, the more sort of copies you have confirming that this is in fact the true sequence. So higher read depth correlates with increased confidence in that sequence. Moving on to the next question, number 23. What is the difference between the adult, egg, and nymph RNA-seq tracks? The RNA-seq data was taken from cells at different stages in development. So for example, they took an egg of a psyllid and then they sequenced it for RNA. This kind of gave a snapshot of gene expression at the time that the psyllid is an egg. At a different stage in the life cycle, they sequenced the RNA for the nymph, and then eventually they did RNA-seq for the adult. So they actually have these 3 different snapshots in time for gene expression. It's like if they did RNA-seq on you as a baby, as a kid, and as an adult. And over the course of a life cycle, you're going to have different gene expression, right? To give an example, hormones change as you get older. Like these things are just common where different genes sort of stop expressing or become more expressed at certain stages of life. Being able to look at the RNA-seq tracks for the different life stages kind of gives you an idea of at what points these genes are expressed. Next question, number 24. What is the difference, if any, between the RNA-seq quantitative XY plots and the mapped reads? So by this, I mean here's the adult quantitative, and then here's the adult XY. So in essence, this is the same data, right? But the XY plots kind of put it into a simpler format to be read. The XY plots kind of show you how things fall, and it gives you an idea of exon boundaries really well. I mean, RNA-seq standard one does as well, but the XY plots are really nice for looking at UTR. So over here we can see a nice gradual taper down to 0. But essentially they're showing the same information, they're just kind of formatted. I find the XY plots very easy to read. looking at these mapped reads, it kind of gives you a better indication of the quality of the RNA-seq data and how much you can trust it. You can kind of get a sense from that as well with the read depth here. Like you can see these are getting pretty decent read depths, like 60-ish. But you can also see that illustrated in sort of the how long these are and like how connected, like how many black lines are connecting between them, and that's also a good indication of the quality of RNA-seq. But they're the same information essentially. Number 25. What are the four most reliable evidence tracks? So this you can find on the evidence hierarchy document, but those are de novo, MCOT, ISO-seq, and OGS 3.0. There are many other tracks in Apollo, but these are what are considered the more reliable ones. And to talk about the other less reliable ones, why would we look at them? So, for example, opening up Apollo, let's say we just want to look at all of these different tracks that aren't them. So like Mikado, Augustus, Snap, all these different things. So they're useful to kind of see variations. So here, it's one location, but we kind of get different versions of the exons. So for example, right here, this snap prediction, it has an exon present here where none of the other gene models do. So maybe if you're looking at RNA-seq data and you see like a spike here where it's showing that there should be an exon, maybe you can drag this model up, cut this exon out from the gene model that's a part of, and then sort of incorporate it into the new one. Sometimes it works, sometimes it doesn't, sometimes it just introduces a bunch of splice sites and gets messy. But occasionally what you can do is kind of get the best version of the gene model by taking bits and pieces from other gene models that you find here. So looking at the different tracks kind of gives you an idea of this. And you can see here, like, these are all ending in different locations, right? These different exons. Like this one here is ending a little bit later than the other ones. So there's just these little differences between the gene models. And even if some aren't as reliable from a data perspective, it's interesting to look at the different versions. See here you have the shorter exons. So by looking at your RNA-seq, comparing it with orthologs, you can kind of use them to get the best version of the gene. And sometimes maybe two models are just as valid. So maybe you have isoforms, which are the same protein, but maybe just spliced differently. So sometimes that occurs as well. So it's important to sort of look at all these different models, like as many as you can look at, the better, and then find the oddballs and look at all the oddballs and see, do any of them look valid, and then see, test out by trying to incorporate some of the different more fringe ones and see if they have sort of any valid part to them. Maybe you can include them and make your gene model better. So here's an easy one. Give me your opinion on the UTR. Looks good, honestly. Here are the XY plots. They're all sort of ending. What I would do is I would zoom in to the end here and make sure that it's aligning perfectly with the longest one and sort of just drag it into place. But yeah, this looks very good. There's a taper. Gene model seems to be ending here. Looks good. Now on this side, in contrast, in our gene model in the annotator box, we have it stopping here, but we see that the RNA-seq taper continues on, and eventually you kind of get a gap here. You can see in the mapped reads, which isn't as obvious in the XY plot, but essentially it would be good to drag the UTR out to about here. That agrees better with the RNA-seq data because of the taper, so you would extend it out. 28. Based on the RNA-seq evidence here, does it look like the gene model should end? No. So we don't see any good taper here, and it's kind of hard to see, but take my word for it. This read depth isn't very good. And you're looking at the quantitative tracks, this kind of looks sketchier. This seems like the sequencing kind of failed us here. There's not there's not a lot of great data. It's just kind of a case where sometimes the sequencing data isn't great. You should just be aware, even if there's nothing you can do about it, you should just be aware that, oh, perhaps there's more of the gene model jutting out over here that I'm missing. So maybe by consulting some of the alternative tracks, you might be able to find something that sort of extends, and you would blast this and see, okay, test some things out. If I make changes, if I extend it, does it blast better? Does it blast worse? And after that sort of process, you can kind of settle on if you think it should end here. But just looking at the RNA-seq data by itself, yeah, you don't see a clean taper or anything. Kind of looks like a dead stop. So 29, rate the RNA-seq data in descending order between these 3 life stages. In adult, we see nothing. So that's the worst, obviously. In the egg, there's some, but a lot of it's not connected. The read depth is very short. It's not very good. And then here in the nymph, it's way better. You have pretty decent read depth. You have them connecting with these black lines in intronic regions, which is always a good thing. And you kind of get this pretty decent picture of this RNA-seq data to where you can clearly see, okay, here's an exon, here's an exon, here's an exon, and here's an exon. This last one here doesn't have great evidence, but nonetheless. So yeah, this is pretty good in nymph. So what do you think this means regarding gene expression? So like I talked before about when I was kind of explaining RNA-seq, based on gene expression at the different life stages, you can tell when a gene is more active. So here, since the nymph RNA-seq is really good, this may mean that this gene is very highly active at the nymph stage, where it's not active at all at the adult stage, and it's somewhat active at the egg stage, which is pretty interesting, in my opinion at least. All right, number 3. So give an example of validation workflow. There are many different ways to sort of approach validation or annotation. Everyone kind of goes about it a different way in a different order. Nonetheless, here's kind of how a normal would go. You'd start off going into the Google Doc, you'd get your sequences, you would look at the copy number table, confirm how many copy numbers you're supposed to find. You'd then go into the 2.0, you'd blat it, trying to find the 2.0 models, sort of examine the different 2.0 models, maybe blast them all, and kind of take a comparison of them. Then you would take, the 2.0 models, blat them at 3.0, look at all the blat hits, kind of examine them. Maybe you want to check to see if certain ones are artifact duplications or not. Then you would just go in and do validation. So you do all the normal steps that you would do when you're examining validation. You're going to get, obviously, very different things each time. Every gene model is kind of like a puzzle. These are some common things that you want to do when you do validation. After you're confident in your gene model, you would write up your conclusion, fill in the comment box, you'd properly name them, put in descriptions and all that. So, but this is kind of I put here more so to kind of get you to kind of formulate your own procedure. Maybe you have a particular way that you like to do certain things, and this order doesn't jive with you. Find what works best and what's efficient for you. So that's been the end of this worksheet. I hope this was helpful and explained some concepts in Apollo for you. Thank you for watching. Have a good day.